My name is Mary Goldman, and this is a demo of UCSC Xena. Just to remind you, Xena is a visualization and analysis platform for functional genomics data. Xena allows you to view public resources such as TCGA, as well as your own data, and I'm going to give you a little flavor of both today. We have a number of showcased public resources on Xena, including the newest datasets from TCGA, the PanCan Atlas, which is what we're going to be looking at today. We also have uniformly processed expression data from GTEx and TCGA, as well as whole genome data from about 3,000 PCOG samples. What types of data does Xena visualize? Virtually every type of functional genomic data. We visualize mutations, structural variants, copy number data, a wide variety of expression types, methylation, phenotype and clinical data, including survival data, signatures, and other classifications. For the demo today, we're going to build on the slides that Jing presented earlier. We were looking at the molecular subtypes in lower grade glioma, and we're going to see if those same subtypes are present in glioblastoma, another brain cancer. We'll be looking at copy number data for chromosome 1 and chromosome 19, as well as mutation status for TP53 and ATRX. I'm going to start our demo by going to our homepage. You can scroll to learn a little bit more about Xena. I'm going to click on the Get Started link here. So when you get here, you'll see our beginner's tutorial. And I'm going to cover a lot of what is in that tutorial, but there's still some things in there that might be useful for you. Scrolling down, you can see a number of live examples that you can click into. And now I'm going to launch the browser. When you first arrive at the Xena browser, we have a welcome banner at the top, along with a number of examples that you can click into. And I'm just going to close this so we have some more real estate. When you first enter Xena, you enter our 3-step wizard to build your very first visual spreadsheet, steps 1, 2, and 3. The first step is to select a study to explore. There are 2 ways to select a study. You can click Help me select a study, in which case you would enter your disease or tissue of origin, or you can select I know the study, and that's what we're going to do. And type in the PanCan Atlas and click Done. We can see here now the PanCan Atlas study. We're looking at about 13,000 samples, so that's quite a few samples there on the screen. Each of these samples is a row, and we're going to be looking at data on those rows. The rows are very compressed, as you can see. One of these bars is about 500 samples. So now we're going to go on to step 2, which is to add some data on these samples. We can add either genomic or phenotypic data, and in this case we're going to add phenotype. We have a number of phenotypes that we suggest that you might be interested in. You can also search for a particular phenotype or click Show All to see everything that we have. We're just going to add the cancer type abbreviation. So now we can see all the different cancer types for each of the 13,000 samples. And now we're going to add our last piece of data. In this case, we're going to add some genomic data, and we're going to add chromosome 1 copy number data. And this is going to take just a little second to draw since it's 13,000 samples for the entirety of chromosome 1. So this is our visual spreadsheet. One row is one sample across all of the columns on the screen and all of the future columns that we're going to be adding. Data is sorted by the first column. In this case, it's being sorted by cancer type, and then subsorted on the column thereafter, in this case, the average copy number variation. We can see that sort order if we move the copy number variation to the front, and now the data is sorted by the average copy number variation for chromosome 1 and then subsorted on cancer type. Moving the column back, we're going to look a little more closely at the copy number variation. Looking at the legend, we can see that deletions are in blue and amplifications are in red. In this case, 0 is copy number normal, and that is in white. We annotate genes to the top, and you can see those in the tooltip here. We can click and drag to zoom in to a particular part of the chromosome and get a better sense of what is happening. And I'm going to zoom out now. So you'll remember that we wanted to view just lower grade glioma and glioblastoma together. We didn't want to see all these other cancer types that we have here. So to do this, we're going to use Xena's find and filter functionality, and that is this search bar up here. This search bar allows you to search all of the data that you can see on the screen. So we'll start by typing LGG, and we can see it dynamically matching my search term and highlighting the samples that do match with these black boxes. And we can see we've found about 500 samples. And now I'm going to add GBM, and again we can see it dynamically matching my search term and highlighting these samples, both lower grade glioma and glioblastoma. And now that we have our samples that we're interested in, I'm going to click on the menu here. There are a number of things that we can do with the samples that we just found. We can filter down to those samples, so only those samples that match our search term are in view. We can clear that filter. We can also zoom to those samples, so all the other samples are there, but we're just looking at the samples that matched our terms. And this can be really great if you want to just get a quick look at some samples, but you're still doing a larger analysis. And lastly, you can create a new column. When you create a new column, the samples that match your search term are in one subgroup, and the samples that do not match your search term are in another subgroup. So this is great for doing a comparison analysis between two groups. So for instance, if we wanted to compare brain cancer to all the other cancer types in the PanCan Atlas, this is the option that we would use. But in this case, we want to just filter down. So we can see here the same trend that we saw in Jing's slides, which is that there are a number of samples in lower grade glioma that have a deletion in the 1P arm. And interestingly, we do not see this same trend in glioblastoma. So we're going to add another column of data. We're going to add chromosome 19, copy number data. And I'm just going to make this column a little smaller so we have some more real estate. And we can see this very prominent co-deletion in lower grade glioma of 1P and 19Q, and we do not see this trend in glioblastoma. So now we're going to add TP53 mutation data and mutation data for ATRX. We draw all of our mutation columns with the gene model at the top. We always draw it in the 5' to 3' direction. We show exons in alternating light and dark rectangles. The coding region is tall and the UTRs are short. And we annotate each mutation to this gene model, as you can see here, and we color them by their functional impact. Scrolling down, we can see what these functional impacts are. Red are deleterious mutations, orange are splice, blue are missense, green is silent, and white means no variant was found. So we can see a bit of what is happening here, but it's a little bit difficult to see with those gray lines. And these gray lines are samples that do not have data. So if we drag this column to the front, we can see here there are a number of samples that are marked as null because they don't have data. So we're going to use that same find and filter functionality to remove those samples. So we'll start out by searching for null, and we can see here that we're matching the samples that do not have data in one or more of the columns. But we want the samples that do have data. So in this case, we're going to add a not equals to the front of this. And now we can see that we are matching the samples that do have data, and we're going to filter. Moving this column back, we can see what we saw in the slides earlier. Lower grade glioma has two molecular subtypes, one characterized by a co-deletion in chromosome 1p and 19q, and the other by mutations in TP53 and ATRX. And we do not see these same trends in glioblastoma. So I want to show you just a few more features on our visual spreadsheet. And the first one is dynamic kaplan-meier survival analysis. You access this by using the column menu at the top of these columns here. And you can see it listed here. I'm going to run the Kaplan-Meier plot just on the cancer type abbreviation. So we can see here the well-known trend of GBM in general having worse survival compared to lower grade glioma. You can download a high-resolution PDF of this image. We also calculate statistics for you. By default, we show the overall survival, but you can look at a different survival endpoint such as disease-specific survival or progression-free survival. We also allow you to enter your own survival cutoff, so by default we show the entire data set, which in this case is about 5,000 days, which is pretty far into the future, so maybe you're interested in the 5-year survival rate, which is about 1,500 days. You can see the graph is redrawn and the statistics are recalculated. And I'm going to close this now. The next feature I want to show you is the chart view, which you get to by clicking here. The chart view allows you to create scatter plots, box plots, and bar charts using the columns from the visual spreadsheet. We calculate statistics for all of our charts, and this can be a great way to get another view of your data. In this chart, we are looking at the average copy number variation for chromosome 1, for GBM and LGG. And what we can see here is that GBM has a higher average copy number variation compared to lower grade glioma, because lower grade glioma has that subtype that has a deletion in the 1p arm. And we can see that this difference is statistically significant. We can also see that lower grade glioma has a much higher variance compared to GBM, and this is again due to that molecular subtype. At the bottom here, we can change the X and Y axis, and now I'm going to go back to the visual spreadsheet. There are two last features that I want to show you. The first is the PDF generator, and this will generate a high-resolution PDF for a presentation or a publication. You can also download the data for your own analysis. You can download the data from just one column using the column menu. You can download the data from the entire screen using this button here. Or if you want to download the entire data set, you can do that using our data sets here. So everything that we've been looking at so far has been on the public data, but I want to give you just a little flavor of what it's like to look at your own data now. To give you a flavor, I'm going to show you just the installation process to show you how easy it is to get Xena up and running on your laptop. So I've already downloaded the Xena Hub here, and now I'm going to double click to start installing it. You can see that it opens a wizard to help you through the installation, similar to Microsoft Office or some other application. And we click Next here, and now we're installing. I just want to say here that we support Mac, Windows, and Linux operating systems. And now we're done. We're going to go to the My Applications folder here. And we can see here is Xena. We're going to click to start it up. And now we're ready to import data. Going back to the browser, you would click on the data hubs here, which will show you all of the data hubs that we connect you to automatically, and you can click on the My Computer Hub, which is the hub running on this laptop. And this is where you would see the data that you had loaded up into it. And there's nothing here right now, but this is where it would be. The last thing I want to show you is on our home page. So if you scroll to the bottom, we work hard to support our users, and we offer a monthly newsletter for people to keep up to date on new features that we are releasing. We also have a public forum and a private email list where you can contact us with any questions you have. You can also find us on Twitter. And with that, I would like to end. I'd like to thank ITCR for the opportunity to demo Xena, and please check us out. Pick up a handout at the back.